Hello, everyone.
This is Jiahui Yang from Sino Biological. So today I would like to talk about some strategies from our companies for making COVID-19 reagents to support SARS-CoV-2 research, diagnosis, and vaccine development. And for this talk, we mainly focus on the antibody development. We will share the information about the decisions and the challenges we made during the development processes. So this talk will divide it into five major parts. First, we'll just briefly go through the SARS-CoV-2 and what is this virus is and what is the key factors about these viruses. Then we will talk about the strategies for making SARS-CoV-2 antibody development, and then we will go through some results and the challenges we have made during the antibody development. For the last two parts of the talk, we will just share some other related tools for the SARS-CoV-2 research and another PROVIR, our viral protein function. As we all know, WHO has characterized the COVID-19 as a pandemic, and these viruses has caused a lot of public health threatening and also affect our economic development a lot. So to study COVID-19, there's a lot of urgent need for the research tools to help the research and diagnosis and therapeutics. This slide is just showing the complete genome of the SARS-CoV-2. This virus has four major structure proteins, M, E, S, and N, 14 non-structured proteins, and other several accessory proteins. So our work is mainly focusing on spike and the N proteins, just as other people did. This is just a diagram showing that what this virus looks like. So we can see that the S protein is on the surface of the virus, and this is a very key protein for the viral entry. It helps to interact with the ACE2 protein on the human cells and help the virus infecting the human cells. Therefore, spike is the primary target for therapeutic antibodies and diagnosis. And for the recently developing vaccines, most of them are targeting spike proteins. And for this protein, I want to add a few more informations. The spike protein can be divided into S1 and S2. There's also a very important domain on the S1, which is the RBD domain. This is the key domain for interacting with the ACE2 protein. The other key molecules we will talk about a lot in the following slides is the N proteins. These proteins is a very sensitive marker when people study SARS, the original SARS viruses. So when we compare the sequence alignment for 7 known human coronaviruses, we found that for the S proteins, the SARS-CoV-2 and the SARS has a very high identity, which is similar for the N protein. So based on this information, we did the following strategies for making the reagents for both antigen and antibodies of SARS-CoV-2. Because there's a very high homology sequence alignment between SARS and SARS-CoV-2, we try to do the first strategies by testing the original SARS antibodies and the SARS antibody libraries to see if we can find some antibodies and recognize the SARS-CoV-2. Because the development speed is very important at this time, if we can obtain the good antibodies or other reagents for supporting the SARS-CoV-2 scientific research, it will help a lot for the following vaccine development and therapeutic development. And the third strategy is start from the beginning. We will immunize the SARS-CoV-2 antigen and then do the antibody development. But the first key reagent we need to have is the antigen from SARS-CoV-2. We will use recombinant technologies for developing the recombinant SARS-CoV-2 antigens. Here are the four major expression hosts available now for recombinant protein expression. And when we go through the information we have about the SARS-CoV-2 spike protein and N protein, we know that the spike protein is highly glycosylated. So here is just a diagram from one reference showing that there are a couple molecules can be modified on the RBD region. We also see that in our results when we using HEK293 cells and the insect cells for expressing the part of the spike protein. We see the molecular weight differences in these two systems because of the glycosylation pattern differences. Therefore, we chose the eukaryotic expression host, HEK293 cells, and the insect cells for expressing the following S and N proteins. So here are several proteins we obtained for after the recombinant expression. We produce the RBD and SECD as one and several the spike fragments. We also successfully obtained the N proteins. When we check their HPLC properties, we can see a high percentage of the one spike component for the proteins. Then we are coming to our strategy number 1 and number 2. We screen the SARS antibodies or the SARS antibody libraries for SARS-CoV-2 antibodies. This is one of the antibodies we obtained from the SARS spike antibodies. This is the rabbit monoclonal antibodies. When we check this antibody, the interaction between the antibody and the SARS-CoV-2 spike proteins, we can see their interaction very well. And then we also retest these antibodies to see whether it can recognize the pseudoviruses of the SARS-CoV-2. When the pseudoviruses infect the ACE2 expression HEK293 cells, we can clearly see the interaction between the antibody and the pseudoviruses. The bottom part of the EM picture is from one of the references, which is citing our products. And this monoclonal antibody products, you can see we got very good EM results, showing that we have successfully obtained one spike antibody which can recognize the SARS-CoV-2 spike. We got pretty much the same results for the N proteins. We rescreened the SARS and N antibody and the N antibody libraries and obtained one rabbit monoclonal antibodies. And these antibodies can recognize the SARS-CoV-2 N protein and also can work for the Western blot applications. Our customers use these antibodies for IHC analysis. We got very good pictures to detect the NP antigen in the kidney tissues. Then we want to check the affinity of these new antibodies. And we found that both antibodies show very high affinity interaction with their proteins by octet. So for the strategy number 3, we want to start from the beginning. We immunize the animals with the SARS-CoV-2 antigens, including the spike and the N proteins. Since we want to obtain the antibodies which can, hopefully, they can recognize the natural viruses or they can have the neutralization functions. So we chose recombinant proteins as the immunogen. We immunize the immunogen in both mice and rabbits to do the antibody development. Fourth developing platform has been used for the new antibody generation. The regular polyclonal antibody platform, the classical hybridoma. We also use phage display library and B-cell antibody cloning techniques for monoclonal antibody generation. Then lastly, we'll test the application validation by ELISA RHC neutralization. We also want to test if this antibody is specific for SARS-CoV-2 or if it has cross-reactivity to other coronaviruses. So this is just one of the results we obtained for these new antibodies. There are 6 antibodies showing that we are testing the interaction by ELISA with SARS spike proteins or SARS-CoV-2 spike proteins at 2 different concentrations of the antigen. And we see that for the antibody number 1, 2, 3, these 3 antibodies can interact with SARS-CoV-2 spike but did not interact with SARS, the original SARS spike proteins, which means this is probably the spike specific antibodies. We didn't show the results here, but we do test these 3 antibodies to see if they can recognize other 5 human coronaviruses, and we found that all 3 antibodies are specific only to the SARS-CoV-2. So we successfully got a very high specific got couple specific antibodies which only recognizing the new SARS-CoV viruses. Then we did the neutralization assays and we found 2 antibodies which can neutralize the interaction between the pseudoviruses and the ACE2 expressing cells. So currently we have a good quality SARS-CoV-2 antigens, and we also obtained several specific SARS-CoV antibodies. So we did a lot of combination and assembled several kits for different areas of the applications. The left chart showing that this is an inhibitor screening kit. So this kit is used for screening the antibody inhibitors, which is for selecting the neutralization antibodies. We also have the antigen detection ELISA kit by one pair of antibodies, and then we can quantitate the amount of antigen in your sample. We also have the antibody titer and assay kit, which help to detect the immunization titer of the antibody you obtain in the samples. Meanwhile, we are still working on selecting more SARS-CoV-2 antibodies, not just for the specific antibodies. We are also willing to obtain some antibodies which can recognize the SARS-CoV-2 and the other coronaviruses. So the study is still ongoing. To summarize of our talk today, and we think we are in highly demand for high quality SARS-CoV-2 reagents. And so this is Sino Biological is working hard on this area. For antibody development, no matter for SARS-CoV-2 or other antibody development, we believe antigen with good qualities is a very key factor. We use several strategies for developing the SARS-CoV-2 research tools, especially for the antibody development. And the first strategy and second strategy, we successfully get the SARS-CoV-2 antibodies from the original SARS antibodies or the SARS antibody libraries. Those antibodies show very high affinity for binding to the antigen. Currently, we are still having a lot of challenges for obtaining the broad spectrum antibodies which can recognize all 7 human coronaviruses, but we are working on that. For now, we have already had several collections of the SARS-CoV-2 research tools, including the antigen and antibodies we have covered in this talk. And we also mentioned we have several data kits for detecting no matter the antigen or the antibodies for the research. This is our last slide. We just want to mention that other than SARS-CoV-2 research tools or other coronaviruses research tools, we have a very large collection of viral antigens, including antigens to influenza, HIV, RSV, and also the antibodies to all these types of viruses to support the viral research. And thanks for your time.